My name is Angelica, and the topic for today is the use of flow cytometry in clinical practice. So, for the background and the intro, the flow cytometry is a complete technology which gives to biologists to study cellular population with high precision. This technology combines sample dimension, data acquisition speed, precision, and measurement multiplicity. A broad range of research applications include immunophenotyping, multiparametric DNA analysis, proliferation, fluorescent proteins, cell counting. How it is used. Thousands of cells per second pass one by one through one or more laser beams in a flow cytometer. The flow cytometer measures scatter lights at several angles and fluorescence emissions. There are three parts that constitute the flow cytometer. The first part is the fluidic system, which permits the hydrodynamic focusing. The second part is the excitation source and optical emission system from the wavelength filters to the detectors constitute the optical part. And the third part is the electronic system digitalizes the signal to be analyzed with specific computer software. So the application of the technique is that the flow cytometry is often used to characterize diseases in clinical settings. Some examples would be like the peripheral blood, the bone marrow aspirate, and cerebrospinal fluid area, all specimens that can be analyzed using the flow cytometry. And this can only be used with the viable cells, which are to be analyzed. It is most commonly indicated for both benign and malignant hematologic process. The measurement of DNA content was one of the earliest uses of flow cytometry. It can aid in several clinical areas, including diagnosis, treatment plans, and monitoring residual or relapse disease. So for the figure data, my experiment question is, can an adipocyte-specific reporter mouse be used in flow cytometry in order to detect white adipocytes? So for my experimental design, I have a adipocyte-specific reporter mice, and this is being used to isolate the TD tomato plus adipocytes. And the TD tomato is basically a bright red fluorescent protein, which is used to express cancer cells. The technique that we're using is the flow cytometry, and the flow cytometry it identifies and analyzes the cell size and the granularity as the cell flows through the device. So the expected outcome when using this technique is that the identification and sorting of the adipocytes based on the FSC/SSC parameters only, differential sorting of large and small adipocytes, and the sorting UCP1 expressing bright adipocytes and detecting heterogenic. So here we have the results on the bottom left. We see that the previous flow cytometry was just the first column. We see that when attempting to detect the AD tomato adipocytes, it was observed that all the TD tomato plus events were saturated on FSC, which renders out the scale. And the FSC basically is a forward scatter plot, and the SSC is granularity side scatter. So for the optimized flow cytometry, when looking at the second column, we see that by successfully increasing the threshold for the FSC detection, it is possible to limit the small events encountered by the machine and thereby allow a more efficient detection of larger events, such as a adipocyte, as seen in the third and fourth column, which if we see here on the top right of the graph, if we look at the arrows on the X, we see that it's basically between the third and the fourth column. The strengths and drawbacks of the experiment is that if we were to use the previous flow cytometry, then the studies failed to detect the majority of the white adipocytes. As you can see on the bottom left side, we don't have any white adipocytes. But when we use the optimized flow cytometry method, we are able to detect the white and even brown adipocytes. So the question that I had is, why is flow cytometry restricted to larger cell types like adipocytes? And here are my references. Thank you.